Welcome everyone to the BrainSpace Initiative. It's my pleasure today to introduce Janine, and hopefully I get the name right. Is it Bisterbosch?
It's close enough. Huh? It's close enough. Yes, it's Bijsterbosch.
Bijsterbosch. Okay, all right, good. Thank you and welcome. She is currently at the Washington University in St. Louis in the Department of Radiology. She's an assistant professor in the Computational Imaging Research Center. And she is heading a lab called the Personomics Lab, which is exploring how brain connectivity patterns differ from one person to the next. And she's looking at sort of connectome, personalized connectome type work. And so she's going to talk today about some sort of large-scale population-based analyses in the UK Biobank data. So very much looking forward to your talk. Thanks for joining us. And without further ado, I will let you take it from here.
Thank you so much. Thank you. It's really nice to be here. Thank you for the invitation. So yeah, I'm excited to talk a little bit more about several studies that we've done over the past 3 years or so since I started at WashU, broadly looking at mental health in the UK Biobank. And so slightly kind of out of order, I want to talk a little bit about the data set to begin with, because it really sets up some of the questions that I'm going to be looking at in this talk. So for those of you who don't know, the UK Biobank is a study that's being acquired in the UK. The full data set is 500,000 people. And essentially in the UK, they have a national health system. And so what they did was they just sent letters to everyone who was within this age bracket of 40 to 69 back at the time of recruitment, saying, would you like to participate in this study? And so that about 7 million letters, I think, were sent, and out of that 500,000 people enrolled. And then there's imaging being acquired in 100,000 of those people, so 1 in 5. And then there's a second time point in about 10,000 people. And these imaging acquisitions are ongoing, so I think they're about halfway with both the first time point and also the follow-up time point. It takes a while to scan this many people. Just want to briefly mention that this is a multi-site study, but it's not a multi-site study in the traditional sense, because these imaging centers were purpose-built from the ground up for acquiring, with the sole purpose of acquiring the UK Biobank dataset. So they're identical scanners, identical procedures, identical sequences. And so the site effects tend to be much smaller than kind of more opportunistic multi-site studies where it's using the infrastructure that already exists. And so this is what they call prospective epidemiological data set, which means that there's not one specific disease focus. The idea is that so there's not really any inclusion or exclusion criteria either. So some of these people might have had, strokes or diseases before they entered the study, but then the idea is they enter the study, they're going to age, and eventually they're going to die, and everyone is going to get followed up until death. And so the prospective aspect here is really important, and it makes the study really valuable. And because it's so big, you should end up having a large enough sample size of many different patient populations that you're interested in looking at. And so they get a full set of clinical records, ICD-10 codes and things like that. And then there's genomic blood markers and lots and lots and lots of phenotypic data. I like to give the example that one of the question is, how much butter, how thickly do you spread butter on your bread? And then people have to kind of say in terms of the thickness. So, and how much cheese did you eat last week is one of the other questions. So there's really, really rich lifestyle and phenotypic data available in this data set. And so I'm really excited about this data set from the perspective of mental health, because I think the way that we've been going about, and when I talk about mental health, I mean quite broadly, we have a bit of a focus on depression and anxiety symptoms, but I purposefully make this quite broad because I think that's one of the opportunities that we have with studying mental health in a data set like the UK Biobank, where in previous work that has been done in this field, which has been super valuable, but when you get funding to study 100 people, you got to be really picky about who you're going to study. You got to, you might want to get medication naive people, or you might want to get first episode people, or really severe and treatment resistant people. Whereas and often comorbidities are exclusion criteria for studies like that. And to some degree, in some of those studies, you end up getting such a kind of tightly controlled sample that looks nothing like mental health looks like in the real world, because there's just such an extreme array of comorbidities and differences in symptoms and things like that. And so I think the UK Biobank is a really exciting opportunity to have a kind of real messy real world sample that is big enough to delve into that a little bit further and to understand that a little bit further. You also have a big enough sample that you can do really statistically robust things like exploratory separating exploratory and confirmatory samples. You can delve into heterogeneity and try to understand that a little bit better. And because there's such a really broad range of different measures that get acquired in the UK Biobank about lifestyle and things like that, you can tap into risk and resilience factors as well. And so this here becomes essentially the roadmap of my talk today. So I'm going to talk about multiple different studies that we've done over the last few years, many of which are kind of relatively hot off the press. So a lot of the work that I'll be presenting is currently under review, or one thing I think is not even submitted yet. And so, yeah, I'm going to talk about each of these 4 topics in a little bit more detail. And we'll start off looking at symptoms and comorbidities. So this is work that was jointly led by 2 of my graduate students, Rosie Dodd and Kayla Hannon. And I want to talk a little bit more about really what this is about finding our way through the different measurements of mental health in the UK Biobank. Because it turns out that in such a big data set that there's a lot of kind of complexities and things to realize, and it took us a while to delve into this data set. So this is the pathway of in-person visits that people do. So there's at instance 0, there's enrollment, there's a lot of different measurements that happen at instance 0 when people come to the assessment center, but that doesn't include any imaging. So that includes having blood taken and lots and lots of other measurements, but no imaging. And then instance 2 and there is an instance 1 in the middle. It isn't done by a huge number of people, so we tend to mostly, and there's also no imaging in that. So I've left it out of this figure here. Then there's an instance 2 and an instance 3 in some of the people going through this. And so the timelines is approximately there. Which ranges a little bit. I mean, I think the pandemic might have increased the time windows a little bit because of shutdowns for future people, but this is an accurate summary at the moment. And then we can look at some of the self-report measures that are available. So when people come physically into an assessment center, they complete on a touchscreen, they do the neuroticism score, which is a measure of personality that's pretty closely related to mental health. They do the RDS4, which is something that we coined, and I'll get back to that on the next slide. And then there's a labeling yes or no probable depression status that got introduced by another paper. I won't go much further into that. And so if for those of you who look at depression and anxiety, these might not be the sorts of self-report questionnaires that you would ideally want to use in a study. There are some other questionnaires. So the PHQ, the Patient Health Questionnaire, is one that might be a little bit more familiar to you, and the GAD, the Generalized Anxiety Disorder Questionnaire, is one for anxiety. So those are available in the UK Biobank, and when we first saw that, we were like, great, we'll use those because those are kind of some of the more familiar tools to the field. But then when we looked into that a little bit further, we realized that the timing of this in relation to the neuroimaging is really inconsistent across people, and can often be many years apart from when the imaging happens. And so this bracket here, this blue bracket that you see on the screen, is an accurate representation for the kind of breadth of when this might take place. And so if you think about it, this online mental health questionnaire is basically there's like an email that gets blasted out to all of the participants. And most of them complete the questionnaire, within maybe a couple of weeks, within receiving this email or receiving the reminder. And so everyone does this in a relatively short, similar time span to each other, but where they are in their individual broader kind of pattern of imaging acquisition and visits to the assessment center can vary a lot. And so because of that, we were really interested in measuring depression on the day of scanning. And so because of that, we introduced the RDS-4, which is the recent depressive symptom scales. This is a straight up table of correlations between these things. And so the green columns are done on the day of scanning, and the blue columns are done at this email blast on this online questionnaire. And so I'll draw your attention. I don't know if people can see my mouse, I hope so. Draw your attention to this number here, which is the correlation between PHQ and RDS-4, which is 0.6. This is not that low, but these measures are essentially measuring exactly the same thing. So this is perhaps lower than you would expect. And then when we looked into this further, it turns out that that is related to this time gap. So there are some people, a smaller group of people, who happen to do both of these measures at relatively similar time points. And so that's the people we include here on the left of this figure. And so you can see the correlation goes up a lot higher, more like 0.9. And then it kind of goes down as you include people who have a larger gap in between these two acquisitions. And so we wanted to use this recent depressive symptom scales because we wanted to look at depression in relation or at the same time, depression severity at the time of the scanning and related to the scanning. So we did a validation study of this. So this was done on Amazon Mechanical Turk, which is a really useful platform to get a lot of questionnaire data really quickly. So we collected data from a new set of non-UK Biobank participants, but a similar kind of age range and demographic range. And we compared the recent depressive symptom scales to two more gold standard or more well-known measures. And so you can see some of the results here. This was a retest study, so we could look at both the correlation across the different measures in terms of the summary scores, but also the stability of the responses from one time point to another time point. And so we were happy with this, showing that this measure that we introduced, the RDS-4 measures, very similar things to these gold standard questionnaires. But this is, so just to summarize, the UK Biobank really does measure everything. If you're interested in something, it's going to be in there. And it's because it's a prospective epidemiological study, right? So because they don't have a single disease focus, they're trying to measure as many kind of phenotypes and traits and states relative to as many different diseases as possible. I've done some collaborations in different fields and hearing and other things, and there's always something that they measure. But the timing can be a little bit confusing. So there's a lot of different kind of add-on studies and online studies that they do that are not easily always relatable to the imaging data. And they almost never, they use relatively few standardized questionnaires. So if you are in a particular field and you have your kind of measure of preference, chances are that it's not in there. And so, and part of that is because they're trying to measure everything, right? So you can't do a 30 item questionnaire for every single thing that you might want to measure. So often it is borrowed from standardized questionnaires, but it's reduced in terms of the number of questions. And so validation is just really important to make the most of this and to make sure that we're measuring the things that we're measuring. Here at the bottom of the slide, I mentioned a couple of really nice papers done by other labs looking at validation for some of the cognitive measures. So just in case that's interesting to some of the people here, wanted to draw your attention to that. So that's the end of part one. It was just kind of it took us a while to get into the UK Biobank. It's a beast in terms of the number of measures that are available. So I'm happy to talk about that more afterwards if anyone has any experience or questions. But I'll move on to the next part, which is starting to delve a little bit more into imaging markers. Really, I'm going to talk about imaging for the rest of my talk, but let's start with the first study that we did. This was actually in the same paper that that the first part was about. This is a really big figure of how we selected our sample. I won't talk you through every single detail in here. But I'd like to highlight a couple of things. The first thing we did here was, so we wanted to do a continuous relationship between severity of mental health symptoms and imaging measures, multimodal imaging measures, rather than like a case control sort of comparison. But we did want to make sure that, because this is not a study that's focused on mental health, we wanted to make sure that we had a good representation that of people who actually suffer from mental health problems, that it wasn't, you know, majority people who did not, because then you might be reducing or limiting your effect size. And so we use this one question, which is, have you ever been to see your general practitioner for, I think the question is anything related to stress or depression or anxiety. So it's really a catch-all for mental health. And so we included everyone who said yes to that. And then out of the people who said no, which is a larger group of people, it's probably at least twice as big as those who said yes, we matched people to the ones who said yes. So we found the same number of people as the yeses, and every person was matched in terms of their age and sex and head motion. And so we combined these two samples together, and then we split it up again into what we call an exploratory sample and a confirmatory sample. Not quite split half, because we wanted to make sure that we're next going to do a canonical correlation analysis, and we wanted to make sure that we had in the exploratory sample a really large number of individuals per regressor that was included in or feature that was included in our CCA. So that was the second thing I wanted to point out, and it's really large. It's really a luxury to be able to do this, splitting up into a completely independent held out sample, and still have these 4-digit sample sizes that I'm mentioning here. So these were some of our results, and this is a huge figure, but I want to draw your attention to a couple of things. So first of all, the numbers in the top right corner. So we found in the exploratory sample, there are actually 2 significant canonical correlations. I'm focusing on the first here because that was the one that kind of replicated the best in the held out confirmatory sample. And so these numbers at the top are the actual canonical correlation. So we did CCA is a way to relate a number of different imaging measures, and we used multimodal imaging measures, so FA, gray matter volume, resting state connectivity, to a number of measures of mental health that you can see at the bottom right, kind of insert here. And so we found a canonical correlation. It was lower in the confirmatory sample, which you would expect because there's going to be some overfitting in the exploratory sample, but still highly significant. And then the red and the blue bars, so these are essentially the weights for a number of individual imaging variables. And the red bars are the weights for in the exploratory sample, and then the blue bars are what we saw in the confirmatory sample. So again, you can see some disagreement, but also some agreement there. I should highlight here that we didn't just repeat the CCA in the confirmatory sample, but we used all of the estimated coefficients from the first one. So it's how well it fits using the coefficients that were estimated from the exploratory sample. And so as an interim summary there, we did find significant and quite nicely reproducible neurocorrelates of mental health. But 2 things I want to point out. I mean, one of the things was this was a huge figure. You couldn't even read some of the stuff, and I wasn't even showing you all of the imaging measures that were relevant. So that's just to say that the results are really hugely multivariate. And even combining all of those multivariate contributions, the effect size that we see in terms of how much explains in relation to mental health is relatively small. And so that kind of leads me into the second half of this part 2 of my talk, which is to talk a little bit about data pollution. Because to us, it was kind of surprising to see that the effect size is so low. We have a large sample size here. We're measuring a large number of brain features. We think that mental health is something that originates in the brain. And so why is our effect size still relatively low? And this was work led by my graduate student, Tai Easley, And the question here was like, there are a number of different ways that there are a number of different aspects of the data that might lead to a reduced effect size. And so we wanted to, one of those, for example, is that you're getting a noisy measure of the mental health phenotype. So the depression score that someone gets is not a perfect true measure of their level of depression. For example, you know, answering in a question of how depressed did you feel over the last 2 weeks, you know, what's a number, what's a response, you know, quite a lot for me might be different for you, and it might even be different for me on a different day when I'm feeling better or worse. And so one of the manipulations that you can do to try and address this a little bit, and this is not perfect, but it helps to average phenotypes across instances. So in the UK Biobank, most of these types of measures are repeated, and for this study we looked at traits that were expected to be stable over time. And so when you average different responses together, you can get a more accurate estimate of the underlying trait because you're averaging out some of the noise. The same goes for noisy brain metrics. So we only have in the UK Biobank 6 minutes of resting state data, for example. So we're going to get a pretty noisy estimate of the connectivity from that, and we can try and improve that by adding, for example, the residuals from the task fMRI data. I'm not going to go into a lot further detail of what we did exactly, but I'm happy to talk about that more at the end. Another challenge and kind of potential source of data pollution is imbalanced distribution. So this is what the distribution looks like, for example, for neuroticism. So most people are low neuroticism, and then there's like this long tail towards the higher scores. That can be a challenge for regression models because it sees much less of the higher scores, and so the regression models tend to be biased towards the more common scores. But so one thing you can do there is actually, and this is again a luxury thing to do in a data set like the UK Biobank, you can just cut it off. You can see how many subjects you have in the very highest score, and then you can just undersample some of those majority scores so that you only select the same number of individuals. So you're going to take a big hit on your overall sample size, but it might help a model to capture all of the scores equally well. And then the last one that we were interested in is population heterogeneity. So maybe there are multiple subgroups within the UK Biobank of individuals who have a different kind of set of brain correlates related to the same symptoms of mental health. And so maybe by splitting the data set up into more homogeneous groups and then repeating the fit within each group, we can find larger effect sizes of the brain basis of mental health. And so we wanted to test each of these options. In contrast to the previous study, we used random forest regression for this study, and we can talk about that more at some point. So these are some of the results here. The line at the bottom where it says Dadi et al. So this is actually a replication study where we use the same code and the same data set, but a larger data set that was published previously. And so the bottom line is not our results, it's the results from a previous study. And the line above that is our results where we have approximately double the sample size. So you can see already that helps a little bit in terms of increasing the R squared, the amount of variance explained. The one above that, interestingly, so the one in the middle on the right-hand side, you can see the ends. So the next one up is the balanced phenotype, and you can see that for fluid intelligence, these are the two phenotypes that we're predicting are fluid intelligence and neuroticism. You can see that for fluid intelligence that we see an improvement in R squared, even though we have a strong decrease in the sample size. Then when we add in additional imaging features, we see an increase again. And then when we combine the added imaging features with the balanced sample size, again, we see a slight increase there. Neuroticism the results for neuroticism are a little bit all over the place, and I don't have a particularly good explanation for that. But so we were encouraged by this, and I think, you know, it's not magic. We still are in very low effect sizes. It's not like, you know, we're suddenly jumping up to a much higher R squared, but we can see that each of these manipulations, at least for fluid intelligence, do make a difference and do kind of seem to linearly add up and help quite a bit. And overall, you know, we're almost tripling our effect size from 0.02 to 0.06 here, which is a pretty big increase, relatively speaking. So to summarize that, data manipulations to address pollution can help to improve results, but effect sizes remain low. It's not a magic solution. One of the things that I learned in the last 3 years working with the UK Biobank data set is that there's a tendency when you have such a huge data set to use what I call chuck it all in approach, which means, you know, use all the subjects that you can get, use all the features and measures that you can get, and just like throw it all in. And one of the things I've realized is that is not always the best approach. So we saw that, you know, balancing the distribution so that the model sees, for example, each score equally often can actually help. And so being thoughtful in terms of what subjects you use, and in some cases what measures you use, can really help more so than throwing everything in. Now, I mentioned at the start of this study heterogeneity, and I didn't really show the results. They weren't particularly convincing in this study, but I want to delve into that a little bit more. So let's go on to part 3 of the talk, looking at heterogeneity in population mental health. So this is work led by my graduate student, Kayla Hannon, and it's been selected for an oral presentation at OHBM. So if you're going to Glasgow, come and find her and us there. And I want to lay out a little bit how we in this study how we were thinking about heterogeneity, because you can imagine 2 different patients. I'm calling them patient A and patient B here. And these 2 patients have the same diagnosis. So they're both diagnosed with major depressive disorder. But then I'm listing the symptoms here. And this is, instead of a game of find the differences, this is a game of find the similarities. So if you looked at this for a little bit, the only overlapping symptom between these 2 patients with the same diagnosis is fatigue, which is a kind of poorly defined symptom to begin with. And so, I mean, these are fictional examples, but this is based on the items in the DSM, the diagnostic criteria of the DSM-5. This is a realistic scenario where these people would get the same diagnosis. And so that's one form of heterogeneity that we call clinical heterogeneity. But let's now compare that same patient A to patient C. And in this case, they have the same diagnosis, but they also have exactly the same matched set of symptoms. Now, it might be the case that the biological mechanisms that give rise to these same symptoms might still be different between these patients. I'm giving an example here, of course, from the overwhelming slide that I showed you earlier. We know that it's probably not one abnormality, but a multivariate kind of pattern that gives rise to these symptoms. But that multivariate pattern might nevertheless be different between patient A and patient C. And so we call that biological heterogeneity, and it's kind of it might be occurring independently of the clinical heterogeneity that we saw previously. And so if this is the case, and again, this is a hypothesis, we don't know necessarily that this is the case, but if this is the case, then maybe what we need to do to kind of tease this apart and disentangle this is, first of all, identify clinically homogeneous groups from our population. And then within each clinically homogeneous group, look to see whether there's residual biological heterogeneity, so whether there are biological subgroups within those groups. And so that was kind of the setup for Kayla's study that we did here. So we, instead of looking for individuals that have kind of shared complex forms of clinical presentation, we kind of look for individuals who isolated one symptom and not the other. So here are three symptoms, three common symptoms of depression: anhedonia, so that's a lack of motivation or interest, low mood, and then somatic disturbance, which is more kind of physiological. In most people who suffer from depression, these symptoms are correlated. So most people, have a mixture of all of these symptoms. But when you have a data set as big as the UK Biobank data set, you have an opportunity to find the rare people who display, excuse me, who display one symptom, but not the other symptoms. And so that's what so out of thousands of people in the UK Biobank, you can see these sample sizes are relatively low, but these are really clean patients who really suffer from only one symptom and not the others. In addition to these symptoms, we looked at some other kind of features of the clinical presentation in the same way. So again, isolating some of these things. So we looked at chronicity, which is kind of the lifetime load of depression. How often have you had depression in your lifetime? Late onset, so these are individuals who only experience their first time of depression after 60, which has some differential kind of etiological pathways related to it. Acute severity, so that is like in this moment, how depressed are you feeling? And then and again, so people who are in the acute severity group are low on chronicity and all of the other things. So we're really trying to isolate each of these different elements that make up what in most people is a mixture of depression. And so you can see that in most people it's a mixture of depression, because this last heterogeneous group is much larger. And so that's anyone else who has depression, but who does not fit into any of these kind of more focused and clean clinical groups. And so why did we do this? Well, so this is trying to tease apart that first step of clinical heterogeneity. So the hypothesis here is that if we now look at some of the brain measures, that we can find larger effect sizes, so bigger effects when we look in each of these clinically homogeneous group compared with that clinically heterogeneous group. And so I'm going to show you some results here. We use normative modeling here. I'm not going to go into too much more detail, but in order to do that, you make use of the large data set that is the UK Biobank data set, and you essentially model each of these imaging measures in the healthy population as a function of age and sex, and then we can see how far each individual patient is that was held out from that model fit. We can estimate a normative deviation for each patient, so how far away that patient is relative to where they should be when you look at a large healthy control population. And so positive normative deviations mean higher FA fractional anisotropy in this case, and lower means less than compared to healthy. And so these are the different groups here, the different clinical groups. And so you can see a couple of things to note here. So one of the things to note is this last bar at the end is the heterogeneous group. And so this result confirms our hypothesis that the effect sizes are larger in each of these clinically homogeneous group compared with a heterogeneous group that's just a mixture of all sorts of people. And the other interesting thing here is that not all of the clinical groups have the same effect. So, for example, we can see the late onset group actually has an increase in FA. And I'll show you some other results here. So we did this analysis on a set of about 90 measures, again, multimodal measures. So you can see fractional anisotropy, white matter hyperintensity, gray matter volume, some resting state measure at the end there. These were the imaging measures that showed a significant result after correction for multiple comparisons. And so you can see there's a couple of things to note here. One thing is that that the fact that at least some of the clinically homogeneous group have a larger effect size than the heterogeneous group remains the case in all of these. And you can also see really interesting patterns that kind of go across modalities. So the FA group has a similar kind of pattern, and then you can see the gray matter volume group has a similar pattern again. And then the next question that we wanted to delve into is to look at residual biological heterogeneity. I'll point out, I'm going to present some really preliminary results here. This is ongoing work, so we're delving into this a lot further and working on this a lot further. But just to kind of illustrate the idea, so focusing in on the acute severity group, what we did here was some clustering. We didn't just, so we did UMAP followed by k-means clustering, of course, and then we tested numbers of clusters between 2 and 10 and looked at the most stable solution there, or the most common solution there in bootstrapping. We didn't just want to, whenever you do k-means clustering, you're going to get k clusters. That's what the analysis will do by definition. And so one of the questions is, how stable are these clusters? So that's what the ARI, or the adjusted RAND index, is showing here, which has a relatively high degree of stability, which makes us feel like these clusters might be, realistic clusters when we're doing cross-validation on them. And then the next thing, which is what we're working on now, is to validate these clusters and see, if we look at phenotypes that weren't involved in any of the definition of these groups, do these clusters differ in a meaningful way in terms of those phenotypes? And there's some interesting suggestion here that these two clusters within acute severity might differ in terms of some of their cognitive scores, which is interesting, particularly in the older sample that we're looking at here, in the context of depression sometimes being seen as a risk factor or even a prodrome for Alzheimer's disease. So this is something that we're excited about, and we're looking at a lot further. And again, this is being presented at OHBM as an oral session, so come and find out more there. So to summarize, individual differences in mental health arise from a lot of different sources, and so we're really starting to think about heterogeneity in the population, resulting from clinical and biological sources, and also maybe even the associations between these things. And the UK Biobank is really an exciting opportunity to try and start to disentangle that. It's the first time that we have a sample that's real world and big enough to develop some of these, or to tap into some of these smaller groups. And so, yeah, we did that with this hierarchical method that combines the hypothesis and data-driven approaches. But then validation is really important. Like I said, k-means is always going to give you something. And so we really want to make use of the measures that we have in the UK Biobank to see, to characterize and validate these subtypes. But I think ultimately what I'm excited about is the idea that we can develop a fine-grained nosology, so that we don't just call patient A and patient B, give them both the same diagnosis, but maybe, call patient A, depression type B11, and patient B, depression type A5, and that might inform targeted treatment. We're a little way off of that, but I think it's exciting, and I think this data set has a role to play in helping us get there. And so with that, I'll finish off with the last and shorter section. I want to talk a little bit about a couple of studies we've done looking at risk and resilience factors. Both of these studies are led by my postdoc, Dr. Wei Zhang. And this study is also has an oral presentation at OHBM. So again, come and learn more there if you're coming either online or in person. The idea here is that we know that physical exercise can be really helpful, either to prevent problems with mental health or to alleviate some of the symptoms with mental health. And so the question was, given that this is the case, do mental health and physical exercise have a shared architecture, both in the brain and in terms of genetics? And so on the brain side, we used canonical correlation analysis again, and we did it separately for mental health and physical exercise so that we can compare the resulting kind of patterns. And on the genetic correlation side, we used genomic structural equation modeling to extract latent factors of mental health and then relate those to in terms of genetic correlations to physical activity measures. I'll briefly go over some of the results here. This work focused on resting state measures and only a smaller number of networks that were cognitive networks that were known to play a role in mental health and potentially physical activity. So these are some of the, on the top here in gray is connectivity between networks, and at the bottom in orange, these are amplitudes, so network strength. So a couple of things to note here. These numbers are the weights for that of essentially the contributions of each of these imaging measures to the CCA. And then on the left is mental health, and the right is a completely separate analysis of physical activity. So these patterns are strikingly similar. They're not identical, but they're very, very similar. And interestingly, amplitudes tend to have slightly higher contributions than connectivity measures. And when we looked at genetic correlations, we found genetic correlations between mental health and moderate exercise and overall exercise, and also sleep duration. This is data from a kind of step counter type watch, so it measured sleep duration in addition to some of these other things. And so the last study that I want to touch on real quick, also led by Dr. Wei Zhang, was trying to look at resilience to mental health. And the way we did this is we borrowed a little bit from the brain aging literature. So we start off training a model to predict, instead of age, predict depression score based on brain data, based on imaging measures. And then we apply that trained model to some held out subjects to predict depression score. So what we get is for each of the held out subjects, we get a prediction from the model of like, this is how depressed we think this individual, the model thinks this individual is. And then what we can do is compare the predicted to the true depression score to look at the gap or the delta. And in most subjects, the gap will hopefully be small because that means that the model is doing a good job. But in some people, there's going to be either an over prediction or an under prediction. And so that means that we can identify individuals with particularly vulnerable brain signatures for mental health, or particularly resilient brain signatures for mental health. And so now what we do is we select the kind of most extreme individuals, both on the vulnerable side and on the resilient side, and we relate the gap, so this quantification of vulnerability or resilience, to a number of different risk factors that might explain vulnerability or resilience to mental health. And so these are some of the results here. This list here is the different measures that we looked at. I want to draw your attention to the bottom one here, which is material deprivation. So this is a measure of effectively socioeconomic status or resources available. And we can see that in the resilience group, there was a relationship here on the right. So, larger material deprivation means less resilient. So the more resources are available, the further people are to the left of this curve, the more resilient they are. Surprisingly, we did not find the kind of mirroring effect on the vulnerable, within individuals with a vulnerable brain phenotype for depression. So yeah, that was really interesting to us. And to summarize that last part, I've shown you that mental health and physical exercise share both neural architecture and genetic architecture, and that neural resilience, but not vulnerability to mental health, is linked to socioeconomic status. And yeah, just to revisit, population data really offers a lot of rich phenotypes to investigate risk and resilience factors. But as always, we're struggling with the trade-off between, you could look at so many factors, but you want to make sure that you have the power to do so, and also the interpretability to then look at the results and make sense of them. So it's whenever you delve into such a large data set, it's a real trade-off between what I mentioned, the chuck it all in versus being a little bit more thoughtful and purposeful about what measures we're looking at. So before I end, I want to put this up because there's some stuff going on in the lab that I haven't really talked about that I'm also excited about. So we're doing some longitudinal studies using some of the longitudinal UK Biobank data to look at state and trait differences. We're looking at some structure function interactions with mental health. And that's coming back a little bit to the work that I did previously, looking at spatial differences in functional organization, and we're tying that into structural kind of spatial organization as well. We're starting to look into mental health in relation to cognition and Alzheimer's disease. And then for those of you who know the work that I've done previously at Oxford that I haven't really presented here, I am still doing some follow-up work looking at brain representation. So we're developing a mapping between different brain representations and a way of comparison between different brain representations. And we're doing some cross-species work to look at the cellular basis of some features of networks, including network overlap. So with that, I will thank my lab, who, as you can see, look wonderful even when they're on Zoom. And I'll thank all of my collaborators and my funding, and thank you very much for your attention. I'd be happy to take any questions.
Thanks much, Janine. That was a great talk. I very much enjoyed it. So for everybody listening, if you have questions, please, you can post them to the chat, or you can just raise your hand if you'd like to unmute and ask a question. I had one, which was, I very much resonate with your sort of chuck it all in versus selectively subsampling. I'm just curious, to what degree do you think, like when you do this subsampling, how important is it to do cross-validation, like out of sample, maybe other parts of the sample or whatever, but, and have you tried that and looked at whether it matters? Because I always wonder how, well, even in the data you have, this can be an issue, but when you're, especially when you're selecting extremes or subsets, you can potentially sort of unanticipated biases can crop in. And so just wondering to what degree you've kind of thought through that, those kinds of issues in the work you've done so far.
Yeah, for sure. We had so when we looked at the extremes, so the last study that I mentioned about resilience, we replicated the results using kind of multiple cutoffs for the extremes to make sure that it was stable there, and it was. So it wasn't like we only found the result at one cutoff. It was kind of stable across multiple cutoffs for the extremes, which I think is a useful kind of validation or replication there. The thing about extremes is that they might be interesting. They're often interesting to look at in the context of mental health, right? These are some of the patients that are probably clinically speaking the most interesting. But yeah, I totally agree that we want to make sure that our results are not just a fluke based on like a decision that we made somewhere along the line.
Yeah.
We were thinking about trying to like have a replication. We actually had a really long discussion yesterday about the subtyping that we're doing, and thinking about whether we could have a held out replication sample there. And I don't quite think that works, but what we did there quite a lot is we did bootstrapping, so kind of thousands of times, getting just a 90% or 80%, and then running the same analysis and seeing if you get the same result, and the same kind of allocation of people into subtypes, sort of thing. So I think there's different solutions. I mean, ideally at the one of the first studies that I presented, where we had the full kind of exploratory sample and then a completely independent confirmatory sample, and I think in some cases, if that's possible, that is definitely ideal, even if it's within the same data set. But I think there's a lot of things that you can do, kind of, testing sensitivity to different decisions that you're making and doing bootstrapping, cross-validation, all of these things. But yeah, I totally agree with you that it's really important. And within this type of data set, you have more ability to do that sort of thing than in a lot of other data sets. And so we should hold ourselves to a higher standard for doing that, I think.
Yeah, no, I think it's it's great work. I think yeah, and again, sort of in these sort of large population-based studies, a large portion of the individuals may not have any signal of interest, right? So I think it is important to kind of look like you're looking through it in careful ways. One more comment from me, and then I'll jump to a question from Martin. But you had mentioned at one point about the 6 minutes of resting fMRI data, being short, and I wanted to just sort of put a plug in to maybe if you haven't played with spatially constrained ICA. We have a study, Marlena Duda here, where she's used templates, component templates derived from large samples that replicated, and then showed that she can get pretty similar results, even down to as low as 2 minutes of resting fMRI data. So just something to think about. I know it takes a lot of time to generate new features from these large data sets, but just sort of a brief comment. Yeah.
That's interesting. And I mean, I think so. If you compare, for example, to the Human Connectome project, where you have an hour of data per subject, 6 minutes sounds ridiculously little. But at the same time, we do have a huge number of subjects. So actually, if you do a kind of a group ICA analysis or other, I've played with Perfimo and other measures, you get pretty good, very stable estimates. We've done split halves on those because you have enough subjects there. So it's really but the estimates of the individual might suffer. And so we're starting to do a little bit of work as well, looking at how to combine, if there's 2 scans available for subjects that are a couple of years apart, kind of how to combine them to get even more signal there. So, yeah. But thanks. That sounds really
sure. Yeah, I mean the nice benefit is you can do spatially constrained ICA on single subjects, right? And so then it's easy to combine across single subjects because the component labels, the component ordering, all that stuff is already available for you. So let me get to a question from Martin. It says, have you looked at potential relationships between depression scores, neurocorrelates, and leisure activities other than physical exercise? He's interested in music, including active participation, mainly for leisure. So a shift in topic here.
Yeah, that's really interesting. So there's an interesting question of the interesting leisure question is has a lot of different answers in it. Like there's some question of like, what do you do for leisure, and it has like answers of I go to the pub, I'm a member of a club or something like that, or and I don't think it has music very explicitly in there. So the short answer is we have delved into a number of different measures, but because we looked at diet a little bit, but I cannot say that we've focused on music very much. So that would be really interesting. I don't actually know exactly what the questions are in the UK Biobank. I'm sure there are some, but it's not something that we've delved into a lot. But it's a great suggestion. Thank you.
Reach out to Martin.
For sure.
Another question from Ashkan says, you talked about data manipulation for increasing effect size, data sampling, I guess. And I assume you are suggesting to use these data methods in different studies. My question is that if we do data manipulation in a study, doesn't that mean the effect size we are getting is biased? Isn't this problematic? Oh, sort of a variation on my question, I guess. But
yeah. I mean, so I think
biased in interesting ways, I guess, right?
Yeah, I mean, you could argue with the other. You could argue that what we're seeing, if we're just using the full sample, that is biased because the algorithm. If we can increase it, then I think that means that there's some bias happening in either direction. I don't know what I don't know that you can argue that one is more true or accurate than another. I mean, it comes back a little bit to there are some discussions in the machine learning literature about data augmentation and you know, how to optimize training for different machine learning algorithms. I'm in the Computational Imaging Research Center here, and they do a lot of, you know, predicting of or localizing a lesion or a tumor or things like that. And so I think it depends on what you want from your model, right? If you want your model to do well at predicting extreme people, which are likely going to be the interesting people, then you want to make sure that the training data sufficiently samples those people in the extremes. So I think when I've thought this through a few times, but I think in most cases, having kind of a balanced distribution is probably, even though that distribution isn't an accurate reflection of the population, it is still giving the algorithm the best chances of kind of doing a good job at predicting all of the different scores that are possible. So, but I think it's a really interesting question, and I think it's something that we kind of have to keep thinking about and keep working on. One thing I will say, so we did the undersampling for the balance distribution, where we kind of reduced the sample. And what I've seen a lot in the literature is the alternative, which is oversampling. So you can just repeat the same subject. So for the scores that are the least common, you can just artificially add those subjects in 3, 4, 5 times, so that they're relatively well represented compared to the other scores. I think that, to me, is more of a problem, because then you're not getting then you might be increasing the representation, but you're not increasing the variance associated with that, because you're essentially just throwing the same people back in. So I think in that case, the oversampling, I think, might introduce more of a bias, because it's just not giving you an accurate estimate of the variance in those categories, in those less common categories. But I think it's a really important topic, and I don't know that there's a kind of one size fits all solution that should be used everywhere. I think the most important thing is to be thoughtful of these things and to kind of consider how what we're doing to our data and to our sample might impact our algorithm fit.
Analytic humility, right?
Right.
Great. Well, thank you so much. We're right a little bit past noon, so I think we'll go ahead and close the session out now. But thank you so much. This was really, really interesting. And it seems like there's a lot more coming, so we're looking forward to it. I think Armin will probably follow up, because we, for the BrainSpace initiative, we'll probably have a Slack channel for this talk. And so if folks want to have subsequent, maybe more detailed conversations or discussions, we kind of open it up and put a place for people to kind of have that occur. So thank you very much. And thanks everyone for joining, and have a great end of the week, you all, and enjoy your weekends. Okay.
Thank you so much.
All right. Bye-bye.
Bye.